Hi, my name is Daniel Kerschensteiner. I'm a professor of ophthalmology and visual sciences, neuroscience, and biomedical engineering. And the mission of my lab is to understand, preserve, and restore vision. Here, I'll focus on the basic science side of the lab and tell you about a couple of ways in which we're trying to understand vision. But first, I want to highlight the scale of the problem, focusing on the retina and its brain targets. The retina is composed of more than 150 different neuron types that assemble into more than 40 circuits, which send different feature representations of the visual world to more than 50 brain areas. And we're trying to characterize the feature representations of the retinal output, understand how they arise in the presynaptic circuits of the retina, where this information is sent in the brain, and how it is converted into behavior. Now, we study the organization and function of the visual system at a variety of different scales to understand both detailed computational mechanisms as well as its overall architecture. On the high resolution end of things, we recently found that while most people think that neurons function as computational units, in fact, different neurites of the same cell can function independently, shown here by 2-photon calcium imaging of the dendrites of an interneuron, where you can clearly see the different dendrites responding selectively to different stimuli. On the large scale end of things, we combine viral tracing with patch clamp recordings and morphological reconstructions to comprehensively map which retinal output neuron types innervate which brain areas. One thing that this revealed was that only a small subset of the retinal output neurons, 9 of the more than 40 types, support binocular vision. Now, it's important to study the organization and function of the visual system in the context of the behaviors it evolved to support. Among these behaviors, we study how the visual system mediates predator evasion and how it mediates prey capture. On the left, you can see results from a study in which we identified a single retinal interneuron that is required for the innate defensive responses of mice to looming stimuli, which mimic the approach of an aerial predator. And on the right, you can see how we record mice hunting crickets simultaneously on 3 cameras. We then use artificial neural networks to track predator-prey interactions in 3D. One thing that this showed was that mice maintain crickets within their binocular visual field during pursuit and capture. Finally, and this is motivated both by our basic and translational ambitions, we recently pioneered functional recordings from the human retina using both surgical samples and donor tissue, and this led to the first functional characterization of the output of the human retina. We're currently building a system with 4,096 electrodes, allowing for massively parallel recordings and putting us in the unique position to comprehensively and quantitatively understand the signals that our eyes send to our brain. So if you're interested in joining us in this or any of the other endeavors, please feel free to stop by during the Meet the Professor session or to reach out to me via email. Thanks.